This is drawing 43, and in this drawing we are going to see how the MHC2 clip gets onto the outside of the macrophage. So let's read our title sentence up here. Now we need to learn about the MHC clip on the APCs, antigen-presenting cells, such as macrophages. We saw how the MHC1 works on body cells, and it gives a little sample of what's going on inside the cell. And now we're going to see how the number 2 clip works. Okay, so we're going to draw a macrophage right here, and in this corner I want to make a little MHC2 clip just to remind us what it looks like. So let's start like this and go blobby. It doesn't matter exactly what this looks like. It's this big blobby amoeba-like macrophage. So the rest of the macrophage is off the page. This is kind of one half of them. And we're going to put numbers over here. We're going to number the drawing, and the words are already there for you. So, you know, we're going to put number 1, and then if you want to know what's going on in the picture, it's listed right here. So the words are right there, but I might focus in a little bit like that. Okay, so we can label this if you want to. You don't have to, but if you want to remember, if you're afraid you won't. Macrophage. So this is a little inlet thing here where these would be pseudopods, little false arms that are growing out around, and they're going to close in on a pathogen. So I'm going to make my pathogen look like a bacteria, and I'm going to make them orange. This picture works all right with just black and white, but if you want to use color, you can. We've been using orange for those little antigen dots, which is why I'm going with orange. Okay, so number 1 is right here, and words say a pathogen gets eaten, and the process is called phagocytosis. The vesicle containing the pathogen is called a phagosome. So let's draw what happens next right here. Now we're going to draw another picture here and here, so keep your things kind of small. So this thing closes in and creates like a little bubble, a vesicle. And there it is inside. Okay, so this is the same thing. This is a time progression. First this happens, then this. And this thing is called a phagosome. Okay, and over here we want to make a lysosome. L-Y-S-O. Some. And remember, a lysosome has little digestive enzymes. It's like the little digesting stomach of the cell. And then these two are going to merge. They're going to bump into each other, join together. So we're going to draw a little arrow here that indicates that they come together. And of course, the neat thing is about anytime you have a vesicle or part of a membrane, when they touch, it's like 2 oil droplets touching. Boom, immediately they join. So these things touch. There's no special chemistry that has to go on. They're just going to merge together naturally. And they're going to create this kind of, we'll make this still a little lumpy so you can see this used to be the lysosome, this used to be the phagosome. So we'll make some of these chemicals leaking over here. We'll put our little bacteria guy there. Could be some other kind of pathogen. Mine just looks like a little bacillus with a flagellum on him. So this thing here, let's see, we'll put this as number 2. And I'm going to write they merge. And this thing is called a phagolysosome. You just put the 2 words together. A phagolysosome. And they make a big deal about this in the textbook, about knowing the name of this when they join together. Okay, so what's going to happen here? These little digestive dots are going to get going. They're going to dissolve the little pathogen and break it down to little bits. You notice what we don't have here is any chemical weapons. Remember, the neutrophil was the one that had the chemical weapons. And so you don't really want to destroy this thing because there's a point to digesting it. We want the little pieces, we want nice little pieces that we can then present on the outside membrane. So we don't use really as far as I know, chemical weapons. We'll just say digestive enzymes. Okay, so we have now a vesicle with tiny bits of pathogen. And these bits can be, no, probably like, maybe a dozen or 2 dozen amino acids long. So that's pretty small, 10 to 20 amino acids. Okay, so these are bits of pathogen. So the pathogen is no longer dangerous. It's been neutralized. Now over here, make a little section of endoplasmic reticulum coming over. ER. And just like in the previous picture where we saw the MHC1 clip being made inside the ER, okay, we're same thing here. We're going to have a little ribosome if you want. Okay, and it's making little protein. And let's see, let's have the MHC2 clip is going to look just like the MHC1. In fact, we can draw a picture of it right over here. I was thinking this would be a nice place to put it up close. Now, of course, it doesn't look like this. It's the real one. It's not really square, right? But this is the stylized way that we draw it. And we draw the little protein in there. MHC2. Remember, it's called major histocompatibility complex. But I always like to think of it as my house cleaning, because you're showing, by presenting these parts, you're showing what's going on inside the cell. Like the cell is the house. Okay, so we want to, problem is here, if you look over here, what happened in our last one? Well, remember there was little bits of protein? We had that little protein shredder and it put little bits floating around in here. Well, there probably are the same thing, little protein bits. There would be little bits of protein floating around in here, but we don't want those little bits of protein on here. We want a piece of pathogen. How can we keep it from getting clogged with a little piece of protein? We want the things that are over here inside this lysosome. So very cleverly, I'm just going to use pink, you can use whatever color. It's got a little safety clip on it. Looks something like this. Okay, it's got a little clip on it that keeps anything from sticking to there, because it's not time to do that yet. So let's see, we have this, oh sorry, we need to put this, this is number 3, where we've got bits of pathogen. Meanwhile, number 4, meanwhile the MHC2 protein is coming out of the ER. Okay, so let's label this down here 4. This is what's going on right here. So we want to get the MHC2 out of there, and the job of the endoplasmic reticulum, it's like one of those parts you're like, what does it do? You know, quick, what does the ribosome do? It makes proteins. What does the Golgi body do? It packages proteins. What does the lysosome do? Digest things. What does the ER do? And you're kind of like, that's a little bit harder, right? Well, it puts things into vesicles. That's one of its jobs. Is it puts things. It can blow bubbles. You know, like when you blow a bubble, bubble gum. You know, you have that thing come out. Okay, you can think of it as like the bubble blowing machine. So it can put this thing. It just bulbs out and pinches off a piece of this. The ER is made of plasma membrane. It's the same stuff, that phospholipid bilayer. That's what it's made out of. I mean, we could have made it a double layer like this to remind us. So if a little piece of it pinches off, that's what you're going to get, a vesicle. A vesicle is nothing more than a little bubble of membrane. So here's our little MHC2 clip in there, and it's got a little safety device on it. Right, and it's probably even sitting in, it's embedded. I'm going to go ahead and show it embedded in one side of the membrane here. Alright, and this thing right here, if you want to label it, that pink thing is safety device prevents ER proteins from sticking. So if you just want to make a little note there, if you think you can remember it, it's fine. So we're going to have another merger here. This one is going to join with this one. And this little safety device is going to come off. Actually, make the two little sticks. Make the two sticks look like they're sitting in the side of this, sort of. Okay. I'm going to put the safety device sitting to the side. And what will happen to the safety device? Well, it's made out of proteins. It's a protein gadget. It'll get recycled, chopped up, made into something else. So the safety clip comes off. And now, yay, because we got there's little bits of pathogen in here. And one of those little bits can stick. Only one. These others get wasted. Yeah, they get wasted. But this process is going on thousands of times. You know, the other, you know, pathogens and stuff are going to be, I mean, it's going to be constantly processing more and more pathogens, more and more bits. So, yeah, we have a little bit of a waste. These ones are going to be wasted. So we got one stuck in there. And this is number 5. The phagolysosome merges with the vesicle containing the MHC2 clip. The safety device falls off and a piece of pathogen protein sticks to the MHC2 clip. Okay, next, it floats over on its way. Here it is. I'm just going to make sure, see how I embedded those two little pieces like in the membrane here, because you'll see what's going to happen. It touches, let's pretend it has already touched. As soon as it touches that's why I draw in pencil, because then if I need to erase, I can. Like that. I'm going to make it a little bit bigger here. So this is the vesicle, but it's touched. It's touched here, so it's starting to join. It joined with, remember that exocytosis thing? This is exocytosis. So here it is in here. There it is. And then it's just going to open up. It's going to open up, and this thing is just going to, the vesicle will actually become part of the membrane. It's made out of the same stuff. So this is number 6. The vesicle merges with the membrane, and then we'll just go to number 7 would be, there it is. The vesicle disappears, it completely merges. And number 7, there it is. So that's how an antigen-presenting cell presents the antigens. And I could put down here, I want to add a note, only on APCs, antigen-presenting cells. That's where you find MHC2. MHC1 is on all body cells. So this guy would have some 1s also. It's a body cell, would have MHC1 on it, but only the antigen-presenting cells. What are the antigen-presenting cells? Macrophages, dendritic cells, and also B cells, although we're not going to emphasize the fact that B cells do it. They do, but these guys are the main antigen-presenting cells that we want to talk about. Okay, so let's go down here and look at T cell receptors. Like this. Okay, T cells have receptors on their membranes that will lock on to either MHC1 or MHC2, not both. Well, this means that there are 2 different types of T cells. And when T cells mature, they become one or the other. So you need both. So we're going to look at the 2 different types of T cells. So on this side, we're going to look at a CD4 cell. So let's make the side of a T cell. We're not going to see the whole thing. It's a round cell. So this is the plasma membrane. And we can draw right here. We can label this T cell. And over here, we're going to see the macrophage sticking out. So this would be the trying to write sideways here. Macrophage. And I think I'll draw, this is the plasma membrane. So we can embed the receptors. Okay, so we're going to look at this MHC2 clip. We just saw it. We're going to aim for having the antigen piece in the middle. So the clip is going to be, put MHC2 right about here. Okay. Here's our goofy piece of pathogen. This is MHC2. And if you want to make, you don't have to, if you want to make it a little color or something, just make it something light. Sometimes they show these as different colors to emphasize that there's 4 proteins joined together, but you don't have to. Okay, so the T cell is going to have something that locks onto here. It has a piece, it's kind of like MHC2, but it's going to look, we're going to make it look like a lot like MHC2, but with more of a Y shape. And then 2 things that stick in the membrane like that. And it has the same 4 proteins like that. Okay. And it's going to feel this little bit of pathogen. And this receptor, it's not called MHC anything. It's actually called the TCR, T cell receptor. And its TCR isn't for anything tricky at all. It's just T cell receptor. Receptor is any little gadget on the side of the cell that receives information. So these are unique to T cells, but I want to show you something. Look at the shape of this, and let me show you. Does it remind you of an antibody that the B cell makes? Right? Remember, the B cell makes these Y-shaped things, and these 2 areas on the tip, this is called the variable region, and this thing here is what matches or doesn't match an antigen. So if you took this section here off of an antibody, and they've colored it differently here, and you kind of stretch these out and put little sticks on them, this and this are almost the same thing, basically. A T cell receptor is almost identical to one little top thing of a Y on an antibody. So this is the area that would match the shape of the antigen. And if you remember, B cells each B cell makes only one shape. So you have like millions of B cells, each making their shape. Same thing with T cells. Each T cell has a receptor that is a unique shape. So you have to have millions of different T cells, each having a different shape, and you hope that maybe one of those shapes will be right or almost right. So let me show you another picture. This shows you how it goes in the membrane. Okay, shows you that there's a really nice little phospholipid bilayer there. Okay, and I just want to show you another way to draw it. It's more complicated. Here we've got, this is the MHC1 here, T cell receptor, and we're going to draw something in a minute called CD8. But I think this is kind of hard. I think using squares and rectangles and stuff's a lot easier, so I didn't really like that. Here's another way somebody drew it. Okay, this T cell receptor, it's got this like clippy area here, but again, I like this drawing, but this is just too difficult to draw for our drawings. So we're going to go with a kind of rectangles and squares theme, and it'll be just fine. Okay, so I'm going to zoom in a little bit here. All right. So this T cell's job is to determine that it's connected to a macrophage. Now you remember, a T cell doesn't have eyes and doesn't have a brain. When this little receptor comes, if the shape happens to match, it goes, oh, there's a bit of pathogen. Well, it doesn't think, oh, but what goes on is it recognizes that little thing. Now, only this part is touching, okay? It's touching here. This receptor only knows that it's touching a little bit of pathogen. It can't see what's behind here, okay? How does it know whether it's touching an actual pathogen? It could be this could be a pathogen. It's just touching the side of a pathogen. How does it know that it's talking to a macrophage? Well, it has another little sensor thingy, and we're going to draw this right here. This thing's going to look kind of like a shish kebab. I'm going to draw a stick, and then there's 4 chunks on the shish kebab. 1, 2, 3, 4. And this side sticks in the membrane, and I'm going to make, of course, they're proteins, right? Little chunks of protein. So it really is like meat on a stick, right? If these are proteins, meat's protein. Okay, we've got meat on a stick here. I'm going to make mine green because it's more friendly. It's not warlike because these are the helper cells doing this. And this is the thing that will recognize. It reaches over here. This is like a little finger that goes, it reaches over and it recognizes the MHC2. It goes, oh, I feel the MHC2. I know I'm connected to a macrophage because pathogens don't have this. I can feel I'm connected to a macrophage. Okay, I am being presented with a little bad thing I need to go get rid of. And then signals will go down. If this matches, then if it doesn't match, nothing will happen. But if it matches, then that'll activate, it'll send signals, this thing will send signals into the cell and activate the T cell. Okay, so this has to match and this has to match, both. And this little thingy here is called CD4. So C is for cluster, D is for differentiation. They started naming these proteins. They're like, oh, a cluster differentiation. It was like a little group. So it was called like little group number 1 and little group number 2. Well, this was the 4th one discovered, I guess. This is little group number 4. So it's just surface protein number 4. Just call it, everyone knows it as CD4. CD4 recognizes MHC2. That's its job. That's all it does, and then sends a signal. So guess what these T cells are called? These guys are technically, their right name is CD4 or CD4 plus. CD4 positive means it does have it. Yes, I have it. CD4, this is a CD4 T cell. The informal name, I'll put in parentheses underneath it, is helper. They usually get called helpers, but correctly, and you'll see both. You'll need to know both names, the helper or the CD4. And that's how it gets its name. If you don't know about this little protein, you're wondering why it's called the CD4 T cell. That's why. Okay, so we have T cell receptor. Let's just put a note right here before we leave this. Point to this end here, and let's say this end is very similar to an antibody, those IgGs, and only recognizes one piece of a pathogen. It only recognizes one shape. Okay, and then we could put a note here what these things mean. These things mean and when it's activated, let's see, these These mean activation. I'm trying to think of what else you're going to wonder. Wait, what was that thing? These mean activating. When CD4 cells are activated, they go and alert B cells. Put and T cells. They go and alert other cells. Okay? When CD4 cells are activated, they go and alert other B cells. We can put and other T cells. Okay, and that's all you need to know about helper cells right now. We'll talk more about them later, but that's what you need to know is that the helpers have this thing. They recognize the MHC2, and then they activate. And of course, once we know, hey, this is the right shape to go after, this T cell, what's he going to go and do? He's going to start doing mitosis and divide, and make a zillion copies of himself because he's suddenly very useful. He's not going to be naive anymore, is he? He's going to know his job, and he's going to start doing it. He won't be naive anymore. Okay, so on this side, let's see a different kind of T cell. Now, this is kind of like a separate drawing here. You don't have to do this, but I want to make sure it looks like separate drawing. Okay, so starting over again, we're going to do the side of a T cell. Here's, once again, we'll label it T cell. And this one over here, though, we're going to do not a macrophage. We're going to do a regular body cell. This is body cell. So let's put the MHC1 on the body cell. I kind of want the antigen about in the middle, so put the clip about like this. And remember, this guy only has one stick. That's how you tell it's number 1. Put our little piece of protein in here. Label it MHC1. So this is a sample of what's been going on inside the cell, right? My house cleaning. This is going to be a little sample of the activity that's going on in the cell. Hopefully, if it's a normal cell protein, then nothing will happen. If it's something bad, T cell's going to have to deal with it. Okay, so on this side, we're going to put a T cell receptor. You know what that looks like. It's going to come in like a Y, like an antibody. Still has these 4 proteins stuck in the membrane. TCR, T cell receptor. You want to color these or not, you don't have to. It's up to you. Okay, same thing. This end is very similar to the antibodies. If you put another note, we can put another one. Each T cell has a receptor with a unique shape. Okay, and that's similar to the antibodies. Okay, I'll leave that. We'll add another note in a minute. Okay, so the receptor latches on here, and what happens if this is a CD4 and it's got this green thing coming across? Nothing's going to happen because the green shish kebab doesn't recognize MHC1. All it does is recognize number 2, so nothing will happen. However, this guy, this is a different kind of cell. This T cell has it's got these 2 long sticks coming out. They're embedded in the membrane down there. And then they have, this is the way they're usually drawn. Of course, it's more complicated because it's a protein. But this is the way they usually draw it, like this. I'm going to make mine red. Red for this T cell's out for blood. It's a killer. We're drawing a killer cell. He's dangerous. Okay. There. So this is going to recognize, right? It's designed to recognize MHC1. And this has a name. It's a CD. This is CD number 8. Well, I mean, maybe it could have been 6 or 7 or 15 or whatever, but in the order of discovery, it's number 8. So this type of cell is called CD8. If you want to put plus, you can, but that's okay if you don't. It means it has it. It's positive for that. CD8 cell. And its informal name is, you can call it killer, and I'm going to put a slash, or you need to know this one too. I like killer, helpers and killers. That's my preference. But recently they've gone to calling it cytotoxic. A cytotoxic T cell. So you'll see TC, that's what they'll say, T cytotoxic. So you need to know that word. All right, so CD8 recognizes MHC1 on body cells. So how does it know that it's not it is not stuck to a pathogen, or it's not on a macrophage? Because this thing only matches up with MHC1. So what happens if this is a match and it's looking for a bad thing? Now, we'll talk more in the next drawing about T cells being trained. They go to the thymus for training, and they are trained to recognize, to not attack body cells. If this was a piece of ordinary house cleaning, oh, this is just a normal house cleaning protein, and this T cell was like, well, I don't like that, I'm going to kill you. And the body cell's going to say, what? It's just a normal, there was nothing. It's like you going, what? I had a sock in my living room. Okay, you can't ship me for that. It's not like I'm making bombs in my living room. I'm not dangerous. Okay, if this is a normal, not dangerous protein, you don't want the T cell killing off body cells. You want this to recognize. But if it is something dangerous, this is a bit of virus laying around, or it's some kind of mutant protein that doesn't belong in there. Okay, the T cell can't fix the body cell. It's sad, but it has to kill it. That's the only way to deal with a problem. You know, you got a lot of cells, you can make more, and so it'll kill it. So let's put down here, if pathogen shape matches this receptor matches this shape, the T cell receptor shape. If the pathogen shape matches the TCR shape, then the CD8 cell will kill the body cell. How does it do it? Well, it's got a little gun called this perforin gun. We'll draw the perforin gun in the next drawing. It's got a little, it just basically shoots this thing that it opens a hole in the cell. It puts a hole in it, and then it can even put toxins in through that hole. So it's kind of nasty. Okay, so for this drawing though, that's all you need to know. This is how the body recognizes how the T cells work, how the T cells make decisions about what to do, and it has to do with these little things, the CD4 and the CD8. The CD4s are called helpers, and the ones that have the CD8 are called killers or cytotoxic. And we'll learn more in the next drawing.